Okay, AP Environmental Science. I'm going to take a few minutes and I want to talk to you about your insect biodiversity lab. I've been working on grading those and it's been very difficult. And there's a couple of things that I have seen that you're having big problems with. One, I kind of knew you would have problems with because a couple of you told me right after you handed it in that you didn't understand the Simpson Biodiversity Index. You said that you knew how to calculate it, but you didn't really know what it meant. Well, since the lab is about biodiversity, I really want you to know what that means. The good news about that is the Simpson Biodiversity Index is not anything that you have to memorize to use for the AP exam, but I do want you to understand what it is. The more distressing thing to me is some of you don't seem to understand very well the scientific method. Now, I didn't take a lot of time on the scientific method early in the class, and maybe I should have. My assumption was...
Mr. Harper, can you please call the front office? Mr. Harper, please call the front office.
Sorry about that. My assumption was that you had covered the scientific method enough in other classes that you would be solid. I want to talk about a hypothesis because in a lot of your labs I saw very weak hypotheses. So here I took this from a website, study.com, but it gives a definition of hypothesis. And it says a hypothesis is a proposed explanation that aims to answer the question formulated. And the hypothesis must be testable through experiment. So for it to be a scientific hypothesis, it has to be testable. But the more important thing to me here is it has to be an explanation. It has to explain something. Now, on your lab, you were supposed to come up with a hypothesis, and I want to talk about what you were supposed to come up with. This was directions straight from your lab. It says a controlled experiment must have a hypothesis. And since our lab was about insect biodiversity, it tells you specifically that the hypothesis has to be about insect biodiversity. And for this lab, our hypothesis had to be about biodiversity in 2 different areas within your local ecosystem. And the hypothesis has to predict a relationship between an independent variable and a dependent variable. For all of you, your dependent variable should be insect biodiversity, because we're making a prediction about biodiversity. Now the question is, what would affect that insect biodiversity? There's a number of different hypotheses that you could use. You may predict that the amount of sunlight would affect the biodiversity. Maybe sunny areas have less insects and shady areas have more insects. You might predict that the amount of moisture affects the biodiversity. That damp areas have more biodiversity, that dry areas have less biodiversity. Those would be acceptable hypotheses. I'm going to use a different example for my hypothesis to try to explain what I'm looking for. And I'm going to say that insect biodiversity depends on the amount of organic matter. But whatever hypothesis you use, for it to be acceptable, you have to have biodiversity. And it has to measure that biodiversity in different areas. And you have to have a variable that changes. That could be sunlight, that could be moisture, it could be organic matter. So here's my sample hypothesis. I predict that insect biodiversity will be higher in areas high in organic matter than those low in organic matter. So I'm hoping that gives you a better understanding of what I want as a hypothesis. And since our experiment is about biodiversity, our hypothesis has to include biodiversity. Well, how would I go about testing that? So here's a description of how I would test that hypothesis. To test my hypothesis, I'll sample 3 areas with different amounts of organic matter. A forest floor, that's going to be the highest in organic matter, a big, thick layer of organic matter, leaves, and other decomposing things on the floor. A lawn, it has organic matter, but not nearly as much because it's taken away. You know, you move it with a lawn, you don't have trees. So there's organic matter here, but not as much as there. And a driveway, which would have very little organic matter. You're basically on concrete or asphalt. So the way I would test that, I would collect insect samples in a 1 square meter area of each of these 3 environments, and I would collect them from the same amount of time, 24 hours, using the same type of traps. I will then analyze the specimens caught and calculate the diversity index for each. So what I can see then is hopefully how the organic matter affects the biodiversity. I'm trying to relate one to the other. I'm trying to explain biodiversity of insects in terms of organic matter. It's an explanation. So hopefully that shows you what I'm looking for scientific method for a hypothesis and dependent and independent variables and how to test them. I now want to look at examples that show how the biodiversity index is used. I'll do that in a second video.